Welcome to VetReview. In this video, I'm going to talk about liver of horse, dog, and pig and how to differentiate between them and ox. So I've already talked about the liver of ox in a previous video. So in this video, we'll start from the liver of horse. The lobes are right, left, middle, and caudate, and unlike ox, the left lobe is further divided into two parts, lateral and medial. Okay, the visceral surface has gastric impression, duodenal impression, colic impression, and cecal impression. In case of ox, we said that there are some impressions, namely the omasal, abomasal, and reticular impression. But since horse, dog, and pig, they are not ruminant animals, they are monogastric, so therefore they don't have these structures. Okay, so they have gastric impression instead, and in case of horse, the large intestine is very big and complex. Okay, so the colic and cecal impression are present, but in case of dog and pig, we'll see that these are not present. Colic and cecal impression are present in case of horse. These are not very two important points because whether you can see these impressions or not, it depends on how you have preserved the specimen. Okay, so if you have fixed the specimen as it is, okay, the entire animal is fixed, and then you do the dissection and remove the liver, then you can see these impressions. But if you just take the liver from the cadaver and then fix it, you will not see these impressions. Okay, so they are not very useful, but good to know. Okay, lobes definitely important, especially the left lobe. Okay, so regarding the lobes, there are two differences. First, in case of ox, the division of lobes is virtual. You can see there's no proper lobation. But in case of horse, you can see fissures. They are not as deep as in case of dog and pig, but they're definitely present. Another thing that I've also, already mentioned, the left lobe is divided into medial and lateral part. Okay, so we have the left lateral lobe and left medial lobe. Okay, the right lobe in case of horse is undivided. Okay, it is not divided into further parts. Then the coronary ligament is present and it has two lamina. Okay, so it has two parts, the left lamina and the right lamina, and each of these lamina will give rise to one ligament. So left lamina will form left lateral ligament or the left triangular ligament, and the right lamina will form the right lateral ligament and right triangular ligament. Okay, lateral ligament and triangular ligament, they are the same thing. Okay, and these ligaments will connect the liver to the diaphragm. Again, important point. So in case of cattle, we discussed the right lateral ligament. Okay, so it has only one part, but in case of horse, the coronary ligament is present and it is divided into two parts, left and right, which is not seen in case of cattle. Then the most important point in horse is that the gallbladder is absent. Okay, so in the liver of horse, there is no attachment for the gallbladder because the gallbladder is absent. You can see there's no gallbladder. While in case of ox, you can see the gallbladder right here. In case of dog and pig, you can see the gallbladder as well, right? But in case of horse, the gallbladder is absent. Then the hepatic duct and pancreatic duct together open side by side into duodenum at the hepatopancreatic ampulla. So in humans and in cattle, the common bile duct and the pancreatic duct together join and form one common duct called the hepatopancreatic duct, which then open into the duodenum. Okay, but in case of horse, the common bile duct or the hepatic duct doesn't join or doesn't fuse with the pancreatic duct. Okay, so they remain separate and open into the duodenum side by side. Again, not too much important. Then let us come to the liver of dog. So the right lobe is further divided into right lateral and right central, or we can say right medial part, and the left lobe is divided into left lateral and left medial or left central part. Okay, so in case of dog, just like horse, the left has two parts, but unlike horse, the right lobe also has two parts. Okay, so that is the difference between the dog and horse. Okay, so in horse, the right lobe was undivided. In case of dog, the right lobe is also divided into two parts, lateral and medial. Again, important point. Then the right central, or the we can say right medial lobe, has a fossa vesicae phallae, which is a depression for the gallbladder. Okay, fossa vesicae phallae is the depression of the gallbladder. Not too much important, but this thing is important. The right central lobe has gallbladder. Okay, so it can be asked, gallbladder is lodged in which lobe? In case of ox, it is present in the right lobe. Of course, right lobe is undivided, so we'll just say right lobe. But in case of dog and pig, the gallbladder is lodged in the right medial, or we can say right central lobe. Okay, so this point is important. You can they can ask you in exam, the gallbladder is lodged in which lobe of the liver. Then the caudate lobe has caudate process on the right side and papillary process on the left side. So caudate process is again present in all four animals, but papillary process is special in case of dog. You will not find the papillary process in pig, horse, and cattle. Okay, papillary process is very special to the dog, and this is actually the main difference between the liver of dog and pig. So the bile duct opens into duodenum, 5 to 7 centimeter behind pylorus. An accessory pancreatic duct may or may not join with the bile duct. Okay, a minor point. So the main points were the division of right and left lobe into further lateral and medial parts. Then gallbladder lodges into the right central lobe. Then papillary process is present on the left side of the caudate lobe. So caudate lobe is formed by two processes, the caudate process on the right side and papillary process on the left side. And at last we have the liver of pig. So just I've mentioned it already that the division of lobe is same as that of dog. So we have a divided right lobe, divided left lobe. We have a caudate lobe and we have a quadrate lobe. Okay. Again, as in case of dog, the right central lobe lodges the gallbladder. Bile duct opens at the papilla on duodenum, about 3 centimeter from the pylorus. Nothing important. The main thing is that papillary process is absent in case of pig. Otherwise, the liver of pig and dog, they are very similar. The lobation is same. The degree of lobation is same. The division of the right and left lobe is same. Position of gallbladder is almost same. The main difference is that in pig, papillary process is absent. Okay. So here you can see that there is no papillary process here. But in case of dog, you can see the papillary process. Okay, so this is it for the liver of pig and dog. Again, there's another picture. You can see this is porta hepatica. We have the gallbladder here. This is the right medial lobe, the right lateral lobe, the quadrate or the middle lobe. They are the same thing. Then left medial lobe and left lateral lobe. And you can see that the left lateral lobe in both dog and pig, they are the largest lobes. Okay. So in dog, you can see how big this lobe is. And in pig, you can see how big this lobe is. Okay. So the largest lobe in case of dog and pig is left lateral. In case of ox, the right lobe is much bigger. Okay. So these are all the differences, the major differences that you have to remember. So this is it for this video. If you found this video useful, then please hit the like button, share this video with your friends, and if you haven't, subscribe to my channel. Thank you.